Welcome, everybody, to Science Under the Stars, our December edition. This will be our last talk of the semester, and then we will be picking up in the spring as well. It also looks like our pre-show never really changed slides from that horse slide. So maybe at the end when we're answering questions, or if Isaac could get it up, if he scrolls through fast enough. We have 4 talks planned for the spring, with our first one in February, and we're actually going to receive a COVID-19 update, which should be pretty interesting to see how things are in February. So tonight we will be hearing from Isaac Miller Cruz. He's a 5th-year grad student in the Hoffman Lab. He and I started together, so it's always fun to see how our research is progressing and watch each other give talks whenever we get the chance. And despite COVID, we've never truly been able to catch up and go grab beers and hang out. So it's nice to finally get to see him give a talk this semester. So I'm really happy to be able to introduce him. He studies behavior, much like I do, but he also studies the neural underpinnings of the behavior. So kind of a neurobehavioral biologist. I know there's an actual term for that, but I can't remember. Isaac, do you want to enlighten me real quick? No,
yeah,
excellent.
yeah, you've nailed it.
Okay, close enough. That's good. So he's going to be talking to us about animal brains in a broad sense. So a bunch of different organisms will probably be covered rather than the fish that he normally studies. So Isaac, whenever you are ready, go ahead and share your presentation. You can take it away.
Great. Well, thank you so much, Kyle, for the introduction. It's great to see you too, and great to see everyone come out today. So I think it'll be a nice talk. Yeah, so I won't necessarily be talking about research, but more just in general about animals and brains and some exciting stories that I think you guys will find very interesting. So, and at the end, we'll have a time for questions to stick around. So please, if you have questions, and I think there also you can write them in the chat as well. So that being said, let's get started. So today I want to talk to you about evolution of animal brains and how they change over time. So of course we know that.
I lied. I'm sorry, Isaac. We have one more thing to announce. So this is being recorded, just so everybody is aware, so you can do your screens as necessary, and it will be later posted on YouTube as well. Sorry, bud. Keep going.
Great. Yes, thanks. Cool. So again, as I was saying, we know that animals come in all shapes and sizes. So I've just picked a selection of them. But just as animals do, right, their brains also come in a wide variety of shapes and sizes. So here I'm just showing a little phylogeny of different animals. Here we've got a whale and a pigeon and a frog over here. And we can see that, the brains, even here I've they're color coded by, similar regions that come from, a common ancestor. But you can see that even though they've got commonalities between, there's a lot of differences, right? There's big changes between them. Before we get started talking about brains, though, I do want to talk about what are brains made of. So just like every other tissue in your body, right, they're made up of cells. Brains and the nervous system have special cells called neurons. So the brain is made up of lots and lots of neurons. And so what makes the neuron so special, right? Why is it so unique to this tissue is that we can send signals from one cell to another, right, from one neuron to another very quickly. And so if we look at sort of the breakdown of what a neuron looks like, so here we've got our soma, which is just the body of the cell, our nucleus, right, which has our genetic material, and then dendrites for receiving that signal. But of course, the main takeaway, right, is this long axon right here. This long axon acts just like an electrical wire. So we can send signals down this axon very, very quickly, right, to other neurons. And you can imagine that if we chain all these neurons together, and in humans, we have over 85 billion neurons, right? We get a really complex network between them. And then finally, one more thing I want to point out, right, is we have these things called myelin sheaths. And just like on a wire, right, you wouldn't have just an exposed wire. We wouldn't want to have our axons to be exposed either. So this is almost like the rubber coating that you have on any of your cords at home. That's what the myelin sheath serves like. Now, of course, the brain isn't just made up of neurons, right? We can't talk about the brain without talking about glia cells or glia, as they're sometimes called. So here I'm showing 2 neurons. Here's one neuron right here. You can see the axon, and then where it's the synapse, right, the connection between that neuron and the next one. Here's another neuron right here. And then we've got, here we're showing sort of 3 different classes of glial cells. So here we have astrocytes, which are really important for supporting neurons and helping, especially with the synapses. We have oligodendrocytes, which are helpful with generating these myelin sheaths, right, to really make sure that signal gets across. And of course, microglia, which are helpful in sort of maintaining the health within the brain as well. So just like when we're going to talk a lot about sort of brain size changing or regions changing size. So I do want you to know that, the neurons, the number of neurons are going to change, right? So, the bigger the brain regions are, right, we're talking about having more neurons there, right? More signals, more information being passed, but also more of these glia, right? So it's not just the neurons, right? We're also adding all these supporting cast members as well. When we look across the vertebrates, we see they share a lot of similarities across their brain. So here I'm showing another phylogeny of vertebrates, right, with fish and amphibians, and this is labeled reptiles, but really it should just be a crocodile, and birds, and then we've got our cat mammal here. And so we can look at sort of differences across these brains or similarities and see how they change over time, right? And of course, here I'm showing the phylogeny and the most common, most recent ancestor, or would be at these different nodes across the vertebrates. So we've got reptiles here, which would contain lizards and crocodiles and birds, and amniotes, which would include the mammals, and then tetrapods, which again would incorporate the amphibians, until we get sort of our full vertebrate class over with fish as well. So looking at these brains, right, one thing that we can see sort of right out of the way is that we can divide the vertebrate brain into three main sections. So we get the forebrain, we get the midbrain, and we get the hindbrain, right? So this is just our classifications. But these three different regions, right, the reason that we can say that they're similar across these species is that they share a lot of similarities, right? And not only in terms of function, but also mainly in structure and development. So how they come about is very similar, which is why we can say that that these brain regions are very similar across these species, even though they're so distantly related from one another. So we're going to talk a lot about sort of brain regions or subregions within these larger regions today. So one thing that's really important that the brain does, right? We all know we have a brain; it's really important. But one thing, right, that the brain does that's a big deal is that it helps us sense the world around us, right? So we can take in information from the world, and the brain is where we're going to process it, and then we can send out the signal to act in response to it. And so one thing we want to make sure we understand is how do animals sense the world around them? Well, you're probably familiar with sort of the core senses that we talk about. So we know that animals have a wide array of senses, right? So we usually talk about our basic five senses, right? We've got hearing, and sight and smell and taste and touch, right? So these are sort of basic ones that we consider, right? When sound comes, right, that comes through our ears and we can hear it, and sight, we take in visible light, and then that information is transferred to our brain. There also are other senses, right, that we don't generally talk about, but things like temperature, right? We all know when something is hot, right? We can all tell when something is hot. If you ever touched a stove, right, you can tell right away when something is hot. We also have a sense of balance and motion, right, when we're moving through space. There's also things like inner condition, right? We can tell when we're thirsty or when we're hungry. And then there's a lot of other animals that can do things like electrolocation, right? They can sense the electricity or magnetic fields around them as well. So there's a lot of different senses in the animal kingdom, and we have a lot of senses as well. But one in particular I want to talk about is how do snakes see infrared? Right? We talk about snakes seeing infrared, right? They can see at night, but what does that actually mean? And we can talk about that. So the first thing we have to do when we talk about infrared is to talk about the electromagnetic spectrum, which is just the range of frequencies that characterizes light. Right? So what does that mean? So when energy radiates or light radiates, it moves in different frequencies and wavelengths. So for example, right, if we have low wavelengths over here to higher wavelengths down here, and we get, there's different types of information, different ways that that's released. And so for example, when we talk about radio waves, right, so that's the signal that your radio picks up, and that's transmitted from radio stations, right? It's again, it's all part of the electromagnetic spectrum. Those are really long, right? So the wavelengths, right, the distance between these waves are the size of buildings, right, down to things like gamma rays, right, which are really teeny tiny. But what's important is that both infrared and visible light are part of this spectrum, right? So our eyes, right, just happen to be able to see this range of the electromagnetic spectrum, right? That's visible light. And in most animals, right, sort of fall within this range. And then there's infrared as well, right? So infrared is just light that's sort of below red, right? Hence the name infrared. But what's, there, it also doesn't, it's not necessarily that we have to take it through our eyes, right? Because just like it's energy leaving, right? Energy leaving is heat. So a lot of times we talk about infrared being heat, which is what we're going to talk about, sort of this thermal infrared light. And so, of course, we all know that snakes have can use the special pit organs to sense infrared thermal radiation. So here we've got this nasty python with a warm balloon, and you can see him. It's in pitch black, right? But it's still able to sort of capture and accurately target, right, this warm balloon, which I think is super cool to watch. So how is that done, right? So snakes, there's a sort of a range of snakes that have pit organs. So we get pit vipers and boas and pythons. So here I'm just showing different examples of snakes with their pit organs around here. Now we're going to talk about sort of two classical models that we're using that's used in sort of snake biology. We have rattlesnakes, which I'm sure a lot of people know, who have, you can see right here, they have one pit organ. So that's the place where they're going to sense that heat. And then I've also marked, right, there's also been marked where their nostril is, right? So it's a separate organ from that. Now the python, as you'll notice, there's a lot more red arrows. So unlike the rattlesnake, which has one pit organ, right, the python actually has multiple pit organs. So we're going to look at that and sort of what does that mean for processing information, right? If we have one organ versus multiple of them. So let's first talk about how, in this case, is our sort of diagram of a snake, right? How do snakes see, right? How does visible light translate it into the brain? So here we've got some visible light coming in from different locations. So here we've got from location 1, right? That light travels down and it hits the retina in this specific location, right? So if the light's coming from right in front of the snake, right, it's going to hit this part of the retina. That information is then going to be transmitted right across to the other side of the snake's brain into the optic tectum. So here we've got 1 marked here. You can see there's a 1 light coming from 1 is going to hit 1, and that's going to be transmitted to the area of the brain that's responsible for that area of the visual field, right? So that 1. Likewise, right, if there's light coming in from slightly behind the snake in this area 4, right, we would see that light being processed in this optic tectum over here in 4. So our question is, right, how are the snakes able to sense something visually? Right, we know they have eyes, and they're also sensing something with their pit organs, right? This thermal. How do they combine that information together? And so we're going to look at how infrared light is now maintained. And again, it's very similar to visual light, right? Because again, it's the same type of information. So we have this infrared light coming in. Again, it's not going to the eye; it's going to these pit organs, which again are very sensitive. We call them somatosensory. So it's almost like very sensitive skin, right? Just like we can sense heat in a broad sense, they can at a very sensitive level sense heat. And so again, this infrared comes in, it hits the part of the pit organ that's, in this one area. That information gets relayed through the brain. And then again, just like with visual light, where information is processed on the opposite side from where it came in on, right? The infrared light comes in and then is processed again in the optic tectum, right, which is the area of the brain that's processing this, but in that area 1. So just like with infrared's coming in from 1, it would be processed here in 1. In 4, it would be processed here in 4. So what does that mean, right? What does that mean if we're our snake, if this is us as we're a snake, and we see, this harsh predator in the distance, right? In this case, my dog. So for one thing, my dog's, a mammal, it's warm-blooded, so she's going to release some heat that's going to be transmitted from here. We're going to say it's going to this place 2, which again is going to go process through the brain until it gets over to this area in the optic tectum. Likewise, right, my dog's, we're going to say it's during the daytime, so there's also visible light radiating or reflecting off of her. And that's also going to be processed through the eyes and the brain until both of these signals are going to converge on this same place in the optic tectum. So what we had is that we're taking 2 sets of information, right? 2 ways that we can localize something, one with infrared, one with visible, from 2 different organs. And we have this area in the brain that's going to process both of this information at the same time, which is pretty exciting. Now, I mentioned that rattlesnakes only had 1 pit organ. So what happens, right, if there's multiple pit organs? So here we've got our python diagram. And so visual light works the same way, right? It's still going to hit the eye, and it's still going to go to the opposite side of the brain. But with infrared, right, what we're going to do is this heat is going to enter through multiple pit organs. So in this case, right, we're going to get sort of a continuous picture of where these things are and which pit organs are responding to heat. So again, if we use my dog example, as we can see, the visual light is still going to be processed this way, but now, right, we're going to use these multiple pit organs to combine the information that we're receiving, which I think is very cool. So what does this tell us about the brain, and what can we learn from it? Well, one, we want to know what's actually happening in this area of the brain. So we can use something called a microelectrode to actually record cells' activity. So we can, here we've got our rattlesnake, see with this nice rattle, and our microelectrode that's going to record the activity of the neurons in that brain region. And so that will tell us, are neurons activating, are they firing, and how do they respond to the stimulus around them? And what we can do is that we can input either visible light here or infrared light here, or a combination, or one or just the other, and see how a certain cell is going to respond to whichever stimulus we want to add to them. And what researchers found is that there's a bunch of different types of cells. So I talked about how there's sort of a location based in the optic tectum of where it's receiving the information from. But what we also know is that there's different cells there, right? There's there's a bunch of neurons, right, in the brain. And so different neurons are going to respond to different things. So the first type of cell that we find are what we call OR cells. These are cells that respond to either visual or infrared or both, right? So here I've got, if there's just light coming in, right, they fire. So this is showing the activity of the neurons. So we can see that they fire a bunch of times. Great. If there's just infrared, right, we're going to see that it's going to fire a bunch of times. And if there's both visual and infrared, right, this cell is going to fire just the same. Great. And then we've got what we call AND cells, right? So AND cells has to be visual light and infrared. So if there's just visual alone, the cells don't fire. If there's just infrared, the cells don't fire. But if there's both, right, the cell fires, which is great because this cell is going to tell us if there's something moving that's giving off heat, and it's going to be very specific. Whereas the OR cell is just saying, there's something moving over there, and it's either something that we can see or something that we can sense through a pit organ. Again, not us, but if we were a snake. And then there's a second class of cells which are enhanced. So in this case, we get infrared enhanced visual cells. And what that means is that normally when there's just a visual stimulus in that cell's area, the cell will fire a little bit. But if there's also infrared, right, it fires a lot more. So you can see there's a lot more lines here than there are here. Likewise, we get the inverse, right? If there's just infrared alone, right, the cell fires, and that's great. But if it's moving and it's giving off heat, right, we get a lot of firing. And then finally, right, we get sort of the opposite of that as well, where we get these infrared depressed visual cells and visual depressed infrared cells, which again are these visual cells. They fire when there's a visual stimulus, right, when things are moving. But if there's also heat coming off, they're not going to fire. Likewise, right, these visual depressed cells are sort of the opposite of that as well. So let's focus on a couple of these, and let's see how a snake can see the world, right? We're going to focus on these OR cells, right, which are ones that fire regardless if there's something moving or if there's something giving off heat. And then these AND cells, which are only going to fire when both things are happening at the same time. And then these infrared depressed visual cells, which are only going to fire if something's moving, right, but it's not giving off heat. So now let's look at how the snake will see the world and how it combines these images together to give it a lot of information about what's going on. So here's from our snake point of view. We've got this mouse right here sort of running across the field. We've got a frog hopping along, and we've got a rabbit that's hidden by this bush. Right, so the rabbit's here, but there's this bush behind it, so we can't actually really see it that well, right? Now, if we look at what would it look like with infrared, right? Well, we know that the rabbit, even though we can't see it well, right, is still going to give off heat because it's a warm-blooded mammal. Likewise, right, the mouse is also going to give off heat. Great. And the frog's not, right? Because the frog's cold-blooded. So while we can see it moving, right, we can't really see it that well with infrared. So together, let's combine these images together. So if we combine these images together, now we can use our different cell types to tell us different information. So for example, our rabbit, right? We can't see our rabbit, but there's heat coming off of it. So our OR cells are fired because we're seeing heat or visual, right? So you've got the heat, but the AND cells don't fire. And these visual cells, right? Again, because they're visual cells, they're not going to fire. Likewise with our mouse, right? Our mouse is running across the field, so our OR cells are going to fire. Great, because it's giving off both heat and visual. Our AND cells are going to fire because again, it's that combination of both of those things. But these visuals, these infrared depressed visual cells, which again only fire if there's just something moving without infrared. So again, right, we've learned there's something different happening over here than there is over here based on which cells are firing. And then finally, we get down to our frog, right? So our frog's hopping along, and we've got these OR cells firing, right? Because we're getting a visual stimulus, but these AND cells aren't, but these visual cells that are infrared depressed are as well. So together, right, you can see how the snake combines all these different images together, right, from these two different senses to get a complete picture of the world around it, which I think is very cool. Cool. So next I want to talk about what happens when a fish can't see. So we've talked about sort of gaining what happens, how do they sort of process information, new information, like infrared. But what happens when a fish can't see? There's a fish species in northern Mexico that has multiple times, if a population has moved underground and they've lost their eyes. So these two fish are really closely related, right? These are the fish that live on the surface stream areas, and these fish within the last million years the populations have moved into these caves. Now these caves are dark and they're kind of nasty, but there's a little bit of food and there's not a lot of predators, so it's great for the fish. But because it's a dark cave, there's not really a need for them to see anything, and there's also not a need for them to be seen. So you get this lack of pigmentation here, and you can also see that they haven't developed an eye. But they are fish, and so here's this fish swimming around in an aquarium, moving along, enjoying its life, looking for food. And what's really impressive is that these fish are actually so closely related they can actually hybridize, so they can actually crossbreed with each other. So not only is this a really interesting phenomenon that's happened, but it's actually happened 3 separate times. So 3 different populations this has happened in, at least that we know of, since we can't go into every cave. So what does that mean, right? What happens when this fish, right, that's lost its eyes, right? It doesn't develop eyes anymore. What happens? So again, here we can sort of compare our 2 species. Well, if this fish isn't processing visual information, right, it no longer has eyes, how do the brains compare with the fish from the surface, right, that do have eyes and do use eyes to see the world around it? So we can look and compare the 2 brains. So this would be the front of the brain right here by the yellow olfactory, and this would be the back of the brain. We're looking sort of down at this diagram from the top. And what you'll notice is that it's color coded by regions that do different things. So again, this would be olfactory, so that would be like smell. Visual is over here in blue. Equilibrium, right, which are these sort of the balance. And lateral line, which is back here, which is these if you look along this fish, you can see the lateral line there. And that's how they can sense sort of movements and things around them. So even though fish, if they can't see, right, they can still sense the water and things moving around them. But if we focus just on the visual system, what you'll notice is that in the surface fish, right, they've got these really large, visual cortex, whereas in the cave fish, right, they've got a really small one. So what happened is that over time, right, in evolution, this fish not only hasn't developed eyes, but also its brain has changed, right? Its brain has changed so that its visual cortex is a lot smaller. So the question is, why would you do that, right? Why would that change happen, right? We'd think that having less brain would be, sort of a problem. It wouldn't be that helpful. Well, it turns out that at least in the surface fish, it's actually maintaining the eyes and maintaining the visual system is really expensive. So it takes approximately 20% of its energy at sort of this surface fish to sort of maintain the visual field and looking around. So you could imagine any fish that was born that didn't have to sort of waste any energy in a cave, with developing eyes, with growing that extra tissue, is going to be at a slight advantage because it won't need to, forage for that extra food and get that extra energy, right? It can focus more on things to help it survive and grow and reproduce, which is really exciting. So this is so what can happen, right, with evolution, where when senses change, right, we can see that this is also reflected in the brain as well. Great. So next I want to talk about sort of how do these sensory systems evolve, and I'm going to focus on sort of one class in particular, but the star of the show is the star-nosed mole, who's very handsome, a little mole, who's got this wild 22 appendage rhinarium, which the rhinarium is sort of the fleshy part of a mammal's nose. So if you've ever touched your dog and they've got that sort of wet spot on their nose, right, that's their rhinarium. So looking at this, you can see in this little video, the star-nosed mole is using its nose to sort of feel around the ground and sort of sense the environment around it. And so you can actually see right here in the middle, right, these are its nostrils. Its mouth is actually underneath that. So again, this is just its nose protruding. You can also notice it's got these really big forelimbs, right, these really big paws that it uses for digging. Because again, this mole is living most of its life burrowing and digging underground. And so it can use this heightened sense with its nose to sort of feel the environment around it. And use it for food, but it's really exaggerated, right? It's really wild. And so if we look at this a little more closely, we can see that there's actually 11 distinct appendages. So if we count sort of from the top, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 on each side, and they're covered with these sensitive little organs called Eimer's organs. So right here, they've put the number of how many of these organs, and what you can see is that this one right here has somewhere around 1,600, right? So it's a very sensitive organ, right? These appendages have a lot of sensation around them. You'll also notice that the appendages right by the mouth, right, these ones in the middle, have a lot fewer of these sensitive bumps. And that's believed to be because that's actually helpful for collecting food and bringing food into their mouth. But these appendages are about, I think it's like 6 times more sensitive than our hands are. So they're super sensitive to the environment around them. What's really cool is if we look in this, we want to look in the somatosensory area of the brain, right? So that's the part of the brain that processes sensory information or touch information. So if this animal has evolved this really intense, sensory apparatus, right, these appendages that are dealing with touch and they're very sensitive, we would expect this to be reflected in the part of the brain that deals with touch, right? That deals with sensation, which is what we call the somatosensory. And if we look in this area of the brain, what we find is that we can actually map each appendage to a different area on the brain. So you can see here in the brain, we can match each of the appendages. So here you can see the count from 1 to 11, and then here again as well. So not only is this animal have this unique appendage growing, but each of these is now, right, they've evolved within the brain different areas that are responsible for each one of these in a distinct fashion, which I think is really cool. Not only that, right, so we can look at we can look within the brain. We're going to look at the isocortex, which is sort of dealing with these sensory inputs. And we can see how the isocortex sort of breaks down for different sensations, right? So I've color coded some here. So again, this is that rhinarium, that wild nose that we just talked about. This is the part of the brain that's sort of responsive to the forelimbs, right? Those giant digging paws. Here's auditory for listening, and again, a very small visual field, right? So it's not spending a lot of it's spending a lot of time burrowing. So it's not using its eyes very much. And then 2 little areas that are dedicated to whiskers, right? So just like most mammals, they have whiskers, but there's not as much of their space is taken up by whiskers because again, remember they have that giant thing on right in front of their nose, the rhinarium. So if we look at this brain, we can say, how do animals invest, their sensory real estate, right? So there's only so much space in the brain, and if we're dedicating, a lot more of it to certain sensory areas than to other, right? What would that look like? So we can make a model of this, right? So here's our anatomically correct star-nosed mole, right? So this is what it would normally look like, and if we magnify different areas based on how much of their the sensory cortex it's taking up. So again, with this rhinarium takes up a lot of space. And now we can see that, with on this animal, right, when we exaggerate the proportions, right, this is what we would predict of, right, this is how much of the sensory cortex is taking up relative to sort of how it is on the actual body. And again, we see these big forelimbs, which, in my mind, right, if they're just for digging, you wouldn't want them to be very sensitive, but it turns out they're actually very sensitive, and they use them for a lot of different behaviors and for foraging and social behaviors as well. Um, so well, what does this mean, right? So we want to look at this within the context of evolution, right? Which means we want to compare it to other animals, right? What do other closely related animals look like? Because maybe they're also investing a ton of, of their sensory brain matter towards the rhinarium, towards the node. So here's 4 different insectivore species. We've got the hedgehog, which of course we all know and love, with its beautiful spines, but it spends a lot of its time foraging above ground. So it's got these nice big, relatively big eyes, as well as these long whiskers. We also get the tiny masked shrew, which is a very, very small animal, which again is spending some time foraging above ground. So we can see its eyes over here. And likewise has these really big whiskers as well, which are a little bit more difficult to see. But also, of course, it has a rhinarium as well. And then finally, we get to the 2 mole species, which spend a lot of their time burrowing. And you can see that they've got very tiny eyes. You can almost not even see the eyes on these 2 pictures. But they've got these huge paws for digging, right? These huge forelimbs for digging. And then these large sort of exaggerated rhinarium with the star-nosed moles that we just talked about has this crazy wild 22 appendages on it as well. So what do their brains look like? So if we look at these, we know sort of each of these animals has a different life history, right? They deal with different challenges in their life. And we want to look at their brains and see what that looks like, right? So how do these brains of these animals compare between them? And so I've used a sort of a color coding scheme to show you the differences, right? So if we look at, for example, here, the visual cortex, we can see that the hedgehog and the shrew have a lot of real estate invested towards the visual system, whereas the mole has very little, and it's almost none. It's hard for scientists sort of at this time to find where the visual cortex would be here. Again, we can see that in the hedgehog and the shrew, they invest a lot of this real estate towards the whiskers, right? This green area here and here, whereas very little of it is within the eastern mole, and very, very little in the star-nosed mole. And then of course our rhinarium, right? Our crazy star-nosed mole, the nose, right? This big exaggerated area that we saw. We can see that in the shrew and in the hedgehog, right? It's actually not taking up that much space compared to sort of everything else going around. So again, we can take these proportions, right? How much of the sensory cortex is taken up by a different sense, and we can map it onto the animals themselves. So this is what they would look like. These are the magnifications that we get. So you can see, for example, again, this is our star-nosed mole with its really exaggerated nose. But if we look at our shrew, right, the shrew has these giant whiskers, right? Because the whiskers, it spent a lot of the proportion of its sensory area on the whiskers. Likewise, in our eastern mole, while we see these huge paws and these really big nose as well, right? This really big rhinarium, right? We also see that a lot of it is invested towards the whiskers, which we didn't really see in the star-nosed mole. And then, of course, with our hedgehog, right, we can actually see an exaggeration with those spines. So there's a little extra sensation from the spines, but there's a lot put into the whiskers as well. So what this tells us, right, is that as animals change, right, we know that they evolve and they change and they adapt to their environments. These patterns are also reflected in the brain, right? We can see these changes in the brains as well, which is really exciting. And so I next want to talk about humans. So we've talked about sort of different animals and different systems and how the sensory change. But one thing people always talk about, is that humans, our brains are so big. So I wanted to talk a little bit about why are human brains so big? So when we look at different mammal brain sizes, so here we've got rats and cats and dogs, right? Humans end up being much larger than other mammals, than our other closest relatives between them. And one of the reasons, right, we know this is that if you did the activity today and made the brain hat within this week, you would actually already probably know this. Oops, sorry, jumped ahead of myself there. In humans, right, we have this exaggerated, this forebrain, this cerebrum. And we can, again, just like in the animals, right, we can actually break down the different regions into responding to sort of different sensory stimuli or to more human specific things like language and reading. And here we've got speech, as well as, again, this would be the touch, right? So all the things we just looked at in the mole, right, those would be reflected here as well. And then, of course, vision and hearing and smell and things like that. So again, these classifications, right, we break them down into these different lobes to help understand how we can sort of map the brain and the different responses and this information that it's processing. But that's all we're doing with that. So again, humans we just saw that they're much bigger brains than compared to other mammals, but what do they look like to other primates? So if we look at other primates, like again, our great apes and our lesser apes, we can see that humans are still really large brains, right? So here I'm showing the volumes of brains of these different primates. And what we can see is that humans have much larger brain volume compared to other members of the primate family. And so one of the things that we know is that bigger animals have bigger brains, which again makes sense. So if we look at this graph, here we're going to see the log body weight. So this is our sort of scaled version of the body weight. So bigger animals are on this end and smaller ones are on this end. And then we're going to do the same thing with brain volume. So the higher the number is, right, the bigger the brain volume is. And what we see, for example, this line right here is birds. And so we can see there's a really nice relationship between how big a bird is and how big their brain is, right? So the bigger the bird is, the bigger their brain is. Of course, there's some animals that will there always be an exception. It's biology, so there's never hard, fast rules. And so you can see the pigeon is actually well below that line. Likewise, we can see that apes are over here, right? They're relatively, large animals compared to birds, and they've got relatively big brains. And then, of course, humans are over here. But I did want to point out, right, we're not the biggest mammal, right? We're not the largest mammal, and we don't have the largest mammal brain. So there's a dolphin there as well on the graph. But let's just look at primates in itself. So again, this is a very similar graph, but this time we're just focusing on primates. So all these dots are just different primates. And you can see that as the primates get bigger, right, humans are one of the bigger ones. Not the biggest, but one of the bigger ones. We can also see that the brain size increases, and we get this really nice relationship. So based on human size, this is where we'd expect humans to be. Right? We'd expect us to be right here on the graph. Again, just if we sort of use this regression and we say, where do we think human brains would be? How big do we think they would be relative to their body? Well, if it's like the rest of primates, we'd expect ourselves to be here. But it turns out we have much bigger brains than expected, right? So you can see that, again, this is a log scale. So this is a big difference between where we would expect our brains to be compared to other primates and where we actually are. And so what does that mean though, right? We saw with the fish before that brains are really expensive to maintain. So in order to have a larger brain relative to our body size, there's sort of two things that we need to happen. One is we need to increase the amount of energy we put in, right? If it takes more energy to make something, we're going to need more energy to do that. And then second, we can also reduce the amount of energy, right? We can change how much energy we're using in sort of long term, right? So that's what we're going to say, what we mean by change the energy allocation. So the first one, right, how could we increase our energy? Well, one is with improved diet, right? So by eating cooked meats that are easier to digest, and especially meats themselves, right, we can really increase how much energy we're taking in compared to an animal that has to spend, all day foraging. Likewise, we can also, help each other out. So if we do things like cooperative breeding and when we're working together, right, we can increase how much energy we put in. And then, of course, we can also stabilize where our energy comes from. So something like agriculture, right, where we don't have to spend all day every day looking for food, really helps us, right, increase how much energy we take in the long term. And then, of course, we can reduce how much energy we're taking in, right? So we can make, right, our bipedal locomotion, right? So standing upright and walking is really energy efficient. And then, of course, we can also change how much energy, we can reduce how much energy we need in the long term. So, for example, humans have the longest sort of childhood within primates. So it takes a human a really long time to become reproductively mature, and it takes a long time for them to grow. And so because of that, right, we also have less offspring, right, compared to something a lot smaller. And so together, right, all these traits together means that we can increase our brain size relative to our body by improving how much energy comes in and decreasing how much energy we're spending out. But of course, that doesn't answer the question, right? This is sort of how this could happen. But why would this happen, right? Why would we want bigger brains relative? I mean, it seems to work really well for all the other primates, right? Why would we want these really, really large brains compared to the other primates? So one possible explanation is the social brain hypothesis. And so this again is mapping out different primates. And we've got sort of relative brain volume. In this case, we're looking at one brain region, the neocortex. And then on the y-axis, we're looking at group size. So how many affiliations do they have? And as you can see, right, the animals that have much larger group size, in this case, just primates have larger group sizes, right? We also see the brain size increase as well. So the idea behind this isn't that, by being in larger groups, we have larger brains, or having larger brains allows us to be in bigger groups, but that ecologically, right, it's much better for us to use social behavior to survive. So being social and sociality helps humans survive. But in order to be really successful in a really complex, large social group, right, you need to, for example, remember all the relationships in the group. You need to identify individuals, remember people that helped you, remember people that are part of your group, and people that aren't part of your group as well. And so a large brain is needed to help navigate, right, these large complex social groups. Now, of course, this is just one sort of hypothesis. It's very popular and well read, but there are other ones that explain it as well. But I thought this one was is very common, and I think is sort of a good idea of sort of possible ways, right, that this could come about. Great. So today we've talked for a bit about sort of how animal brains come in different shapes and sizes, and we've talked sort of about different species and how they change over time. But I think it's important to remember, right, that a lot of these animals have brains. I've had people ask me plenty of times saying things like, "Oh, I didn't realize that fish have brains," and yes, they do have brains. And there's a lot in common with our brains as well. So by studying animals outside of humans, right, we can learn a lot about ourselves and about animals around us by trying to understand sort of what's going on in brains of other animals as well. And then I just want to sort of briefly summarize what we talked about today. So we talked about how different animal brains differ and how they change over time. We talked about how do snakes see infrared and how they process that information and use it in their world. We talked about what happens when fish can't see, when cave fish go into the caves and they lose their eyesight. We also talked about how do sensory systems evolve? So what happens when animals evolve and diverge with different environments, when the moles and the shrews and the hedgehogs, and how is that reflected in their brain? And then finally, we talked about human brains and why they're so big and sort of reasons behind that. And of course, I want to say special thanks to Science Under the Stars, an organization that's very near and dear to my heart, and been happy to be part of for the last 4 or 5 years, whatever it's been. Also to members of my lab, so the Hoffman lab, and then of course Nemi for being the best model dog I could ask for. And with that, I'd love to take your questions. Thank you.
You hear me clapping, Isaac?
I can, thank you.
That's excellent. Good. All right, excellent. Well done, buddy. Well done. That was awesome. All right, so we will open it for questions. What I will do is go to the chat first, and then if we get a little bit of dead air, feel free to speak up and ask a question, and we'll go from here. So I saw there was one asked a little bit earlier. Excuse me. So do humans have the largest brains? And if not, why are we the most intellectual species? So I think you might have covered that a little later.
Yeah, I'm happy to answer that one real quick. So we do not have the largest brains, right? So there are animals that have bigger brains than us because there are bigger animals than us, right? As we saw, that body size and brain size go together. I would not say we're are we the most intellectual species? Yeah, we have a lot of really great things that go for us, right? So we have really complex language and abstract ideas that are really great. But I would also say that, a lot of species don't need that, right? There's some very successful animals that don't have that, and they're perfectly capable of what they're doing. And then likewise, right, there's a lot of things that me as a human, right, couldn't do, right? I could not catch a warm bag like a snake could.
Not with that attitude.
Yeah, it's one really great advantage to have this really large brain. But there's some other features I think I could also say that would be useful too. But great question.
Excellent. And also, I think somewhere along the line we were looking at infrared detection with snakes. So, why being able to detect infrared spectrum was evolutionary useful for snakes, but not useful for humans?
Ooh, that's kind of the yeah, my last point.
Yeah.
Yeah, that's a great question. So, it didn't arise, right? We didn't evolve with that adaptation, but it turns out that if you're hunting a lot of predators that aren't moving in the daytime, right, or maybe it's near dusk, like what rodents do, it can be really useful to not have to rely on light to capture them. As well, the snakes, I want to also point out, they don't just use infrared for predation; they also use it for things like thermal regulation. Um, so there's a lot of features in that as well. But yeah, we didn't need it. We were, as primates, we've been very successful, and that was not something that was not a pressure, a selective pressure that we ran against. Unfortunately, because that would be really cool, I think.
That would be sick if we had infrared tech in our brains.
Mhm.
I'm going to combine two questions because they both have to do with animals and they're relatively short. So, why do dolphins have such big brains, and how big is an elephant brain?
Ooh, so I don't know how big an elephant brain is, but big. Yeah, we can come back and look at this slide here. Which shows here the dolphin brain, right? So what we can see is, again, right, we always see this trend that bigger animals have bigger brains. Now, it's not exclusive, right? You could have a very large animal that doesn't have a relatively big brain. As we can see, for example, with the pigeon, right? The pigeon kind of falls off, and the rat kind of falls off too a little bit. But yeah, the dolphins evolved a really big brain, and that, right, is an adaptation that they have, right? So there's a couple hypotheses. I'm not well versed in it, but things like they have really complex social groups, right, which would play into that social hypothesis, right? That they interact and they have these big groups and they have to remember each other. They also have, they communicate with each other in these languages. So having a larger brain apparently is very helpful to a dolphin. And so that dolphins that had bigger brains, right, have were really successful. Yeah.
All right, maybe Gabriel will have to elaborate about this, but can you tell us about dog brains? I'm not sure which direction to take that though.
They have brains, yes. Um, yeah, dogs, I mean, just like most mammals, right? So I think I have a cat brain. Actually, I think there's a dog brain. Here's a dog brain right here. So this is Canis familiaris, right? This is our domestic dog. And this, of course, is the cat, if you want to look at it in comparison. So they have brains. They're, very typical of a mammal and a carnivore. So they process, again, a lot of the sensory inputs, sight, smell, sound, all those things as well.
I actually just got a little bit of word. One of our background analysts looked up the elephant brain. Apparently, they weigh around 12 pounds, that just seems absurd to me. So
Wow,
that's pretty amazing.
that's very heavy.
Yeah.
Which is useful if you're a big elephant. I guess you can carry that around.
So we're wondering how the energetic cost of maintaining eyesight compares to the other sensory systems that have evolved like those of a mole. That's
Oh,
awesome.
that's a great question. I don't have a figure for that, but yeah. So, um, how much energy do we spend sort of with our visual system? So, it turns out that for visual animals, so something like that surface level fish or ourselves, we spend a lot of time on visual, right? Visual is sort of one of our main sensory inputs, especially as humans. So there's a lot of energetic costs to maintaining a visual system. Now, again, it depends on the species, but for us particularly, we do spend a lot on that as well. But in those fish, for example, I think the number again was like around 20% of its metabolic energy. So, which is a big chunk, right? Which makes sense, right? If you're navigating a two-dimensional world that way, you would want to make sure that you do that well, because if you miss something like a predator coming up, right, and that was how you detected them visually, that would be a major disadvantage. So it's good to keep that system up and running. But again, it's not exclusive, right? So animals, for example, the blind cavefish, right, they don't spend any energy on that at all, right? And they also don't spend energy when they're developing, right? So as those fish are growing, right, as they're developing from eggs, they actually start to grow eyes, right? So they start the very beginning of development, and then depending on which population, because as I said, this has happened multiple times, um, that shuts down, and so they start developing eyes, and then they stop, and they stop developing. So they actually have they have the genetic code to develop eyes, but then another part of their genetic code is sort of shutting that down, so they don't waste any extra energy, making valuable eye tissue.
Quick follow up to that because it just got posted. So are you aware of any examples where a sensory system transitioned, I guess similar to the cave fish, and lost its eyes, but then transitioned back to then having eyes? Or do you know of anything similar?
Hmm. Um, sort of an on and off switch. I don't know of an on and off switch, but I like that idea. Now I know that they've induced eyes in the cave fish, so they were able to actually develop eyes in the cave fish. So that's pretty cool. Sort of to cause experimentally so eyes to develop, but yeah, I don't know of any examples where it kind of went on and off and then back on again. It's an interesting thought process, right? Because as soon as a system, for example, like eyes, aren't like once you stop developing eyes, let's say, now you can develop a bunch of eye mutations that won't have any effect on you because you don't have eyes, right? So you can change the genes that control certain features of eyes. So if it's somehow you started after enough time, right, you would start having little changes in the eyes because again, it doesn't matter, you don't have eyes. So then if all of a sudden then eyes were turned back on, I would imagine that all these mutations would make your eyes really ineffective, right, to start out with. Yeah, I don't know. Great question.
Yeah, it'd be pretty crazy, right?
Yeah.
All right. So, what do the mitochondria in the human brain prefer to produce its energy?
What do the mitochondria in the human brain prefer?
So, I think that is asking how do the mitochondria get their energy?
Yeah, so I mean, they have mitochondria, right? Just like every other cell in our body do. Most cells anyway, and they process, just like the other cells process using things. I mean, they produce ATP and using oxygen. I mean, it's been a while since I've looked into. I don't know what the exact question is, but if you're talking about brain food, right? It's just like the rest of your body, right? If you're eating healthy and good for you, whatever is healthy and good for you is going to be good for your brain too.
Way to throw in that FDA endorsement.
That's right. Lots
Uh,
of fruits and vegetables.
how much of brain development is determined by epigenetics rather than strictly DNA? Yeah, have fun with that.
Yeah, great question. So epigenetics, which is a complicated topic in itself, is named above genetics, right? That's actually in its name. So with genetics, right, the idea is that we have in our genes that we've gotten information from our parents, half the information from our mom, half from our dad, and we use that to sort of make ourselves, which are these crazy monsters that have genes, and those genes code for proteins, and those proteins are what do everything in our body, right? So that's sort of the canonical pathway. But with epigenetics, right, the idea is that without changing what's in the genes, right, we can change what's happening within our body. So without changing the actual genetic code, right, affecting sort of outside of it in different mechanisms, we can change what's happening. So what does that mean, right? What that means is that there's still ways for our environment to affect what's happening, right? And that's what we can use epigenetics for, as sort of this interaction, right? It's how we can respond to the environment. So if you're a developing brain, and you're exposed to some chemicals or something in the environment, then that can really change what happens within your brain as well. Now, it's not going to change you from like a a dog-sized brain to a human-sized brain, right? That's probably too dramatic of a change, but it can definitely affect what's happening in your development as well. Complicated question, great question.
All right, I think this is a very specific question in reference to some of the fish that you were talking about. Were they two different species or the same species?
Yeah, so I can go back to that slide as well. So it's actually they're how we define species is complicated, but they're actually right. They're a separate population, right? So a population can be part of a species. So they've actually gone down into the caves, and they can actually interbreed. So we actually get hybrids between the two. So depending on which population they are, some of them scientists I think have labeled as species and others have not. But if you just use the concept of can they have fertile offspring, right? Can they reproduce together? They can. And they do, right? So we find in nature there's sort of these hybrid fish that are maybe have like slightly less eyes, or slightly smaller eyes are sort of starting that degeneration as well. So, but yeah, there's definitely different populations within them as well, but it's very close. That's in my mind, it's a fine line.
And we are out of questions in the chat, so feel free to unmute yourself or ask for permission to be unmuted, and we can put you on live with Isaac.
I will say you can get these blind cavefish at like Petco. I have a couple on my desk at work. They're very nice, and they kind of just swim around. It's fun to watch.
All right, Isaac, dive into your special research interest for a minute.
Yeah, I can definitely talk about my special. So sort of related. So I actually study a fish system as well, Acetilapia bertoni. And they are fish, they're cichlids from Africa, from the Great Rift Lakes. And so my interests are, how does the brain respond to social behaviors, right? So we've talked about the brain and taking in information, but one type of information, right? It's very different to, you can take in visual information of just, some animal running by, but it's very different when you take in information about, for example, another similar animal, right? Another of the same species. So for example, right, when we see another person walk by, right, that's very different to us, and we respond very differently than when we see a squirrel run by. And so my interests are in reproduction. So how does the brain respond to reproductive opportunities, right? When you're a fish and you're given an opportunity to reproduce, right, how does your brain regulate and control that? And so that's what a lot of my interests are. And looking at, when I say response, right, we're not looking at so much the neural activity that we showed before with the snakes, but the gene expression, right? So how does the gene expression change in response to these reproductive opportunities? And then I also have some work looking at the hypothalamus, which is a brain region that I didn't talk about today, but it's really important for controlling a lot of different things, including social behavior, right? It plays a really key role in controlling social behavior. And so I have a project looking at sort of how does this change and evolve across animals, right? So if this region is so important for social behavior, right, we would expect that it might not change that much, but it would change in very specific ways depending on the species. Just like we can see the visual systems and things like that, they don't change that much, or they change a lot in size, but we still need them to see, and there's sort of a certain way that we can go about seeing the world. Again, the hypothalamus, right? We'd expect that there's a lot of things that are really conserved, right? That a lot of animals sort of use the hypothalamus, and we have a common ancestor that had a hypothalamus, and so the question is, how has it changed in different species, and then how does it, again, my specific interest is how does it change in reproductive context? So how does it change if it's breeding season and you're out of breeding season?
Awesome. Before we go any further, Isaac, would you mind putting up the second slide of the pre-show that I sent you so we can get the schedule for next semester up there?
Yeah, of course.
So people can take a look at it. And to what extent is there a difference in neuron density between species and not just like a size and weight comparison?
Yeah, great question. So we talked at the beginning of the talk. I think I can put it. How's that?
Well, we can't see anything.
Oh, so not great. Not great. How? There we go.
Fantastic. Thank you.
Yeah. So I talked a lot about brain size changing, but as we spoke about before at the very start of the talk, right? Brains are tissue, and they're made up of neurons, and also, of course, glia. And so the question is, right, are the brain sizes changing because there's, so many more neurons, or is it, a shifting between, it's like the total number of neurons in the brain isn't changing, and we're just sort of allocating different numbers to different areas, or that there's, that there's actually just, we could have the same size space and just have more neurons packed in there. So as with most things in biology, it's a little bit of everything, right? There's a lot of different options, and it kind of depends on what system we're looking at, which I know is not a super satisfying answer, but it's the truth.
Oh boy. I feel like you can answer this 1,000 different ways, but are there any particular unknown mystery concerning the brain that you hope we will be answered someday?
Oh my goodness. Is there any? Yeah, I think if I like my pie in the sky, right? What's the thing that so we always talk about, what's happening in a cell, right? We say, oh, this cell is activating, or for example, in gene expression, right, we're saying, oh, this cell expressed a bunch of genes. But a lot of times we have to look at it sort of after the fact, right? And so there's some really great sort of new techniques that are coming out where we can actually look at what's happening in the brain in real time. And as someone who studies social behavior, I study reproduction in fish, right? So you could put a fish and you could have another fish sort of swim by it, but it's much more meaningful, and there's a lot more context to it if, right, these fish are freely behaving and reproducing. And so there's some really great work coming out, especially in rodents and mice, where they can look at the brain and see in real time what's happening, right? How are different cells responding, and what's happening in those cells while they're interacting with each other, right? While they're moving about and interacting, which is really exciting. And then on top of that, right, as someone who studies a lot of gene expression stuff, right, so we've talked about sort of the neurons firing, but when they fire, when they're responding to different things in the environment, they also change what genes they're expressing. And so being able to see which genes are changing in real time would be, I think, really, really exciting. And it's something that, we're not quite there yet, but anyway, that's my sci-fi future, is to see what's happening in the brains in real time while they're doing complex, social behaviors.
Any other questions? Oh, amazing. Is your dog's name the Greek word for leg?
Oh, so actually, that's an excellent question. I'm very happy that it was asked. So she's actually named after a lizard species called Nematophorus uniparens. They've actually, since the name has actually been changed to Astilopithus uniparens, but it'll always be Nematophorus uniparens in my mind. And Nematophorus is actually greave wearing, so it's like like shin guard wearing, right, in Greek. And the reason that she's named that, it's one of my favorite lizard species. They're all female, which is wild, right? There's no males. It's all female, so they're parthenogenic, which means they clone. But what I think is really great about them is that they go through a reproductive behavior. So they'll act as a male of a lizard would act, and they'll act as a female would typically act in a lizard, but they don't actually need to, right? They can clone. So what that means is that just like all brains, right, their brains, they're actively, going through different behaviors that we again would say would be male typical or female typical within the same animal, which I think is super cool. And then also my father did a bunch of research on that when I was growing up, so it was a really species that means a lot to me, and that's why she got named after it. And I'm always glad to plug them because I think they're super cool.
Excellent. All right, so what part of the brain processes social status?
Oh, that is a complicated question. So I'm going to make a caveat that we've talked about all this stuff, and And when we talk about something like social status, or really anything that the brain's doing, it's not doing it in isolation, right? It's never really one brain region dealing with, right? We talked about, the optic tectum processing visual information. Well, it's going to interact and respond with the rest of the brain, right? You're not using just one part of your brain at a time. And so something like social status is actually, again, you're going to use a lot of your brain to do that. And there's hypotheses, one that came from my lab, from Dr. Hoffman's lab, called the social decision-making network, which is a bunch of brain regions that are conserved across different animals and have been shown to be responsive to different social situations and also reward systems. And so in that sense, we use what we call networks, right? So it's a bunch of brain regions interacting together. But again, right, it depends. So if anyone says something like what I said earlier, the hypothalamus it's really important for social behavior, but it is by no means an island. It is not working on its own. There are a lot of processes coming in and out, and again, social status is so complicated, right? Because you've got to know the other individuals around you, and you've got to know your rank, and you've got to know their rank, and of course that can change, and you've got to take in what is the other animal doing, and that involves taking in sensory information. So you can see that it gets really complex really fast. So it can be easier maybe to focus on certain brain regions at a time that are really important or key nodes in a network. But there's a whole suite of the brain, and I always like those. I think there was a movie like Limitless, where it was like you could activate that missing part of your brain, and I think that's really funny because if you're not using your brain, like, you have to use your whole brain, right? So if you're not using your whole brain, you're not using any of your brain, which I always liked.
And a nice follow-up to this. So do animals with more complex social systems have larger brains as well?
So that's the idea with the social brain hypothesis. Now, I kind of just focus on primates because it works. It was developed for primates, really humans in particular, and it works really well in humans. But there's a lot of work showing that it's not necessarily applicable to every other species. So, we talk a lot about sort of eusocial insects, right? So these social insects have really complex social systems and castes and things like that. And their brains aren't that big, right? And there's and maybe you don't necessarily need that much information to know sort of your position, right? You can just know, like, I am of this level or I'm of this level. So it's a little more complicated than I think. That's the social brain hypothesis. I think works really is kind of nice for primates. I'm not sure it really applies to other ones as well. And then again, as we've talked about, brain size is sort of one metric, right? There's a lot of things going on in the brain, and is it the full brain that's growing? Is it certain brain regions? I think it's more complicated than that. But yeah, I would say that if you have really complex social systems where you have to remember a lot of faces and a lot of individuals and how they're interacting with you, you're going to need some level, right, some brain capacity to do that, right? More than maybe some an animal that doesn't need to do that. But there are also other things that play into large brains, right? So foraging, right? So if you're an animal that has to remember different locations and different types of foods, and they change over time, those types of things, well, yeah, you're going to need a brain to remember that as well. So there's a lot that can play into it. And then, of course, size, right? That's another big one that we've talked about too.
You were just talking about eusocial insects as well and the size of their brains. Do you mean relative to body size?
Yes. Yeah.
Yeah, I figured. Yeah.
But they are very small.
Yeah. Yeah.
But also they've also there has been some work on, I don't know super well, but a lot of the work that's sort of challenging the social brain hypothesis is can come from insects or I think birds as well. Yeah, it's another one where they sort of found things that kind of rub against what we would predict. So.
Hi, if it's okay, I do have going kind of along the lines of the eusocial insect thing.
Great.
So would you say that it's like looking at a solitary bee species or something like that compared to like a honeybee, their brains would be kind of similar in size?
Okay, yeah, that's a great question. Yeah, thanks for asking that. So, as you talked about, right? So the one hypothesis would be, oh, well, they're really, social. They've got to interact with a lot of individuals. But that doesn't mean the solitary bee isn't doing a lot too, right? It's probably got to travel and remember where food sources are, and it's probably not all that solitary. A lot of animals, right, that we say that they're solitary, but they actually do interact with right, they are social to some degree, especially if they need to reproduce. So our prediction would be, right, again using the social brain hypothesis, that yeah, right, of animals that lived in similar environments and ate similar types of food, which we say is sort of similar ecological environments. Yeah, we would predict maybe the ones that have more social or more complex social relationships wouldn't need a bigger brain, right? But you know, we can't say for sure, right? So it's something we'd have to go test out. And I've never personally studied huge social insects, so I don't know the exact. So maybe someone already knows that, and it would be a great thing to look up and see. A great question.
Have you satiated our audience? You may have.
I'm glad I can answer questions that I am not qualified to answer.
We've been doing it the past 5 years.
Yeah.
Excellent. Well, thanks for coming, everybody. And as you see, our schedule is already filled up with some really cool stuff for the spring. We will be virtual again. UT is all virtual now, so we will be abiding by those guidelines. I think February will be a really interesting time to see how COVID has, I guess, flourished or diminished. And we'll get a nice update from Emily Javin as well. So unless there are any more questions, thanks for coming. Thank you, Isaac, and everybody have a great break, a safe winter, and we'll see you all in February.
Thanks, everyone.